Hello everyone, myself Dr. Subhendu Sarkar. I am an assistant professor in the Department of Chemistry of Brainware University. Today we are going to talk about a very important and fundamental topic in chemistry, namely the fluorescence spectroscopy. So let's begin. So what is fluorescence spectroscopy actually? So this is the process of emission of light by a molecule after absorbing electromagnetic radiation. It is a very fast photophysical process, like it occurs within a few nanoseconds of time. And the most important feature about fluorescence spectroscopy is that it is a highly sensitive analytical technique. And from the perspective of application, it is widely used in vast and different uh, branches of science like chemistry, biology, environmental science, medical diagnostics, etc. So, quinine is the first known fluorophore molecule which stimulated the development of the first spectrophotometer. Now, as far as the molecules are concerned, generally aromatic molecules exhibit fluorescence spectroscopy. So, why these aromatic molecules are uh, showing fluorescence spectroscopy? Because these are the molecules which have highly conjugated pi electronic structure. So this conjugation leads not only leads to the stabilization of the molecule, but also decreases the energy gap between the highest occupied molecular orbital called the HOMO and the lowest unoccupied molecular orbital called the LUMO. And because of this reason, these molecules are showing fluorescence spectroscopy. So, so what are the basic principles of this fluorescence spectroscopy? So different molecules absorb radiation in the UV or visible light. So this is very important. The molecules absorbs in the UV region or in the visible region of the electromagnetic radiation. We can use light source like a UV lamp. So after absorbing this energy through radiation, the electrons of the molecules moves to an excited state, and when this energy source is turned off, the electron loses some energy through vibration and return back to the ground state. So during this process, the molecules emit lights of longer wavelength, that is in the visible region of the electromagnetic radiation, and it shows fluorescence spectroscopy. So as we talked earlier about the uh, like pi conjugated system or aromatic molecules, which shows fluorescence spectroscopy. So here I have shown uh, some fluorescent molecules which shows beautiful color in fluorescence spectroscopy. So you can see all these molecules are highly conjugated pi electronic system. So these are like carbazole, fluorescein, body pi, coumarin, pyridine, quinine, anthracene, etc., riboflavin. So these are the molecules which shows fluorescence spectroscopy, and they have enormous applications in various fields of chemical and biological sciences. Now, let us discuss the photophysical processes which are taking place when a molecule absorbs lights in the UV or visible region. So this is the diagram which is called the Jablonski diagram. So this diagram schematically represent all the photophysical processes happening during the when the molecule is absorbing light and it is going to the excited state. So what is this diagram is all about? So the Jablonski diagram is used to illustrate the absorption and emission of light during various molecular processes which occur in the excited state. The absorption of radiation promotes the molecule from the ground state to a singlet excited state first, like S1, S2, S3, etc. The excited molecules collide with the surrounding molecules, and during this process it releases some amount of energy through a radiationless process called the non-radiative decay, where it releases the energy via heat, and the part of the molecule basically which is responsible for fluorescence is called the fluorophore molecule. So we will come back to the diagram uh, when we are going to discuss various processes I have mentioned here. So at first we will be discussing the non-radiative process, or the process through which the molecule dissipates some amount of heat to release the excess energy. So the first type of non-radiative process is called the vibrational relaxation. So this is a very fast process where an excited molecule drops to the lowest vibrational level of its current electronic state by transferring excess energy as heat to the surrounding solvent or gas molecules. So here simply the uh, molecule drops down from a higher vibrational energy level to the lower vibrational energy level of the same state. Okay, so then comes the other process called the internal conversion. So this is also a non-radiative transition between two electronic states of the same spin multiplicity. So this is very important. So here the molecule simply goes from, say for example, from S2 to S1 or S1 to S0, and the molecule's electronic energy converted into vibrational energy, which is subsequently lost as a form of heat. So here the transitions is happening only between the two same spin or singlet spin state. Now the other one is the inter-system process. So as the name suggests, so this is an isoenergetic non-radiative transition between states of different spin multiplicity. As for example, the molecule is coming from going from singlet S1 to triplet T1 or T1 to S1, etc. So since it involves a change in electronic spin, it is a very it is relatively slower compared to the other two process like vibrational relaxation or internal conversion, and it is only made possible by spin orbit coupling. So now let's move on to the radiative processes, or the processes where the excess energy of the molecule is emitted as a form of light. The first one is our process of interest called the fluorescence. So this is a very rapid spin allowed radiative transition. It typically happens within a few nanoseconds, where a molecule drops from the lowest vibrational level of the first excited singlet state to the ground state S0 via the emission of light in the visible region. So this is very important. Here we will consider only the transition from the lowest vibrational level of the first excited state S1 to S0. And the other process, which is called the phosphorescence, here the molecule molecular transitions happens from the lowest vibrational level of the triplet state T1, say for example T1 to the ground state S0. So in this case also, like as in case of inter-system crossing we have seen, that is also spin forbidden. So this phosphorescence process is also a spin forbidden radiative transition process, and it happens in the region of like milliseconds to seconds. So compared to fluorescence, phosphorescence is a relatively slower process, since here the transition is spin forbidden. Now let us take a quick view about the instrumentation used for the fluorescence spectroscopy. So the instrument used for fluorescence spectroscopy has 5 essential components. Namely, the first one is the light source, which will provide radiation for excitation of the molecule from the ground state to the excited state. Then a monochromator, which will select the wavelength of the radiation for excitation. The third one is a sample cell, where we'll put our sample of interest or the compound for which we are going to like observe the fluorescence spectroscopy. And the fourth part is a monochromator for analyzing the emitted light. And the last one, or the fifth important part of this fluorescence spectrophotometer, is a photosensor to measure the intensity of the emitted light. So here I have given a schematic diagram of the instrumentation used to perform the fluorescence spectroscopy. So this is the light source, and this is the excitation monochromator, and here is the sample cell, where we will put our sample for which we are going to observe the fluorescence spectroscopy. Then there is an excitation monochromator, and here is the detector, which will detect the fluorescence signal of that particular molecule. Now let us discuss the various application of fluorescence spectroscopy. As I spoke earlier, fluorescence has vast and enormous application in various fields of biological, chemical, and environmental and medical sciences. So to summarize, in biological sciences, it is used for DNA sequencing, analysis of various kind of protein, cell imaging. In medical diagnostics, we use fluorescence for detecting cancer. So here the fluorescence technique which is used is called the laser induced fluorescence spectroscopy. For immunoassay and for disease biomarkers. In pharmaceutical industry, fluorescence spectroscopy is used for analysis of various drugs, drug-protein interactions, and also in the process of quality control. In environmental sciences, fluorescence is used for monitoring the water quality parameters, detection of pesticides and heavy metals in various water samples. In food industry, it is used for analysis of vitamins and the detection of food adulterations and for various quality assessment. So thank you very much for listening to my video. Thank you all.